Due to several reasons such as the climate change, the global agricultural direct yield loss is estimated up to 40%, and minimizing such loss is therefore crucial for future sustainability of global crop production. The discovery of RNA-mediated gene silencing mechanism has improved our understanding on gene expression and their regulation. The expression of gene can be easily controlled with use of RNAi or RNA interference technique, which aims to reduce or eliminate the production of a protein from its corresponding gene. The central dogma is the process by which the instructions in the DNA are converted into a functional gene product, which first proposed in 1958 by Francis Crick, the discoverer of the structure of DNA. We are now going to learn a cellular mechanism that uses the gene's own DNA sequence to turn it off, a process that researchers call silencing. Instead of directly editing DNA or inhibiting the transcription process, the key idea behind gene silencing is intervening in gene expression prior to translation. How? By designing a molecule that can specifically identify and disturb the translation process. Andrew Fire and Craig Mello were investigating how gene expression is regulated in the nematode worm C. elegans. They were awarded the Nobel Prize in 2006 for their great finding, a method to silence genes using double-stranded RNA molecules. There are several kinds of RNA molecules in the world. In both prokaryotes and eukaryotes, there are three main types of RNA molecules such as messenger RNA, ribosomal RNA, and transfer RNA. Or we can categorize them as coding and non-coding RNAs. We are focusing on the double-stranded RNA or dsRNA, which are non-coding RNAs that signal for a gene-specific silencing of expression in a number of organisms. Discovered a little over 2 decades ago, small interfering RNAs or siRNAs and microRNAs or miRNAs are non-coding RNAs with important roles in gene regulation. The major difference between siRNA and miRNA is that the former are highly specific with only one mRNA target, whereas the latter have multiple targets. Let's learn more. siRNA-mediated gene silencing mechanism. Long double-stranded RNAs are cleaved into short 21 to 25 nucleotide small interfering RNAs or siRNAs by a ribonuclease known as dicer. The siRNAs subsequently assemble with protein components into an RNA-induced silencing complex or RISC complex, unwinding in the process. Activated RISC then binds to complementary transcript by base-pairing interactions between the siRNA antisense strand and the mRNA. The bound mRNA is cleaved, and sequence-specific degradation of mRNA results in gene silencing. MicroRNA or miRNA is found in most eukaryotes, including humans, and predicted to account for 1 to 5% of the human genome and regulate at least 30% of protein-coding genes. miRNA are small, evolutionarily conserved, single-stranded non-coding RNA molecules that bind target mRNA to prevent protein production by one of the two distinct mechanisms. The miRNA gene is transcribed by RNA polymerase, and it is modified by DROSHA and exported to cytoplasm, converting it to a precursor miRNA, which is cleaved by dicer to make it a mature miRNA. It incorporates into the effector complex RNA-induced silencing complex or RISC complex. Then, the miRNA functions as a guide by base pairing with target mRNA to negatively regulate its expression. The level of complementarity between the guide and mRNA target determines which silencing mechanism will be employed. Cleavage of target messenger RNA or mRNA with subsequent degradation or translation inhibition. picture shows both siRNA and miRNA mediated gene silencing mechanisms together. If it's the siRNA mediated gene silencing, it will result in sequence-specific degradation of mRNA. But if it's miRNA, there could be two results such as cleavage of target messenger RNA with subsequent degradation or translational inhibition. RNA interference is a highly conserved mechanism throughout taxonomical species. In addition to having antiviral activity, RNAi is also believed to suppress the expression of potentially harmful segments of the genome, such as transposons, which might otherwise destabilize the genome by acting as insertional mutagens. RNA interference technique has provided a way to control pests and diseases, introduce novel plant traits, and increase crop yield. Using RNA interference, scientists have developed several novel crops that are currently being used in agriculture. The mechanism has focused on many applications in biomedical research. Some are treatment for HIV, viral hepatitis, cardiovascular, cerebrovascular diseases, neurodegenerative disorders, and cancer, making RNA interference an important tool that makes our lives better. Hoping to meet you with another very interesting topic. Thank you.